Hi, my name is Darshan Kulkarni, and I am here to actually answer questions today. I was recently told that people would be interested in hearing just basic questions and would love to hear our answers, um, basically on how the drug development process works and how drugs do what they do, um, and a variety of other related small questions and answers that healthcare providers or pharmaceutical professionals may know the answers to, but that knowledge has never been shared. My goal was really to provide a forum for answering those questions. Um, as is obvious, I'm not trying to provide legal or medical advice. I'm really trying to just uh, provide specific information that I may know. It may not necessarily apply to what you're doing, but I figured you should at least hear what my opinion is at this time. The question we're answering today is, what is the process of drug development? Drug development is typically thought of as a process that takes millions of dollars. It goes into, well, it takes a long, long time. The idea would be that you go from a concept all the way through getting the product onto the market. What we're going to discuss today is, what is that process? Um, the first thing to remember in that process is that you first have an idea. That idea may come from something as simple as, oh, you saw a research paper, all the way through to you found something that looked really interesting, all the way through an indigenous population, say in rural Zimbabwe, rural India, rural China, whatever, has been using a type of herb for hundreds of years. We don't know much about how the herb does it. We just sort of know that these people use it. Um, so at that point, you might start doing what's referred to as preclinical testing. What preclinical testing really is it's quite simply a way to test if this potential drug would do what we expect it to do. Specifically, would it, when exposed to, let's say, tissues or to just pieces of organs, produce a result that you expect. We aren't going to get into the details. This is supposed to just be an introductory concept. Um, you'll start doing first tests in tissues, and you might even start actually at a computer level. Just run them through machines to see if they do what you expect. Then you might go into tissues and into organs, and then if you see effects, you will try to potentially do that in animals. Now, there's obviously a lot of discussion as to whether animals are necessary or not. The truth is, it's even if you try things in test tubes, it's often almost impossible and impractical to show what the real body would do. There's so many complex organs working with each other, and for that reason, you sometimes have to avoid sticking to the computerized models. That's not to say that models in all situations would be inappropriate. It's just that in many situations you still have to rely on animals. I tend to like animals, and I feel bad about doing something like that. I've never done that, but I would imagine that the reason you have to is because there are no alternatives, or there aren't sufficient alternatives. Uh, Once you do these tests and you start testing things like stability or sterility and process control, you would then get into going to the FDA and saying, look, so what we finished at this point was the preclinical phase. Um, you then go to the FDA and say, look, I have an idea, I have a concept, I would like to start testing this in human beings. So when you are trying to test this in human beings, the first thing you would do is tell the FDA, here's everything I have. Do you think I could go and try this potential drug in human beings? You file what's referred to as an IND, or an investigational new drug application. At that point, um, you wait for the FDA. And once you wait for the FDA, hopefully the FDA will either come back to you with questions, or if you don't hear back from the FDA, then that means that you are free to go and you're free to start your studies. At that point, you would do what's referred to as a phase 1 study. A phase 1 study is often the most problematic phase. because you know almost nothing about what this drug would do, this potential drug would do in human beings. This is your first exposure to human beings, also referred to as first in man studies. And the reason you do these studies is because you're doing this in very few people. I hesitate to call them patients because you aren't treating anything. You're actually just testing a product. And the goal here would be to see, okay, are we really hurting anyone in any severe way? That's why you have processes like informed consent, where you tell them that we don't know much about this potential product. We're giving you things with the idea, with the hope that it'll work, but we don't really know. These are healthy subjects, people who don't actually have a disease typically. There are exceptions, but we're talking primarily about typical things. Typically, they tend to also be men because you want to avoid the cycle fluctuations. Women have natural monthly cycles, and you want to avoid the cycle fluctuations because you don't know whether the cycle fluctuation caused something or the drug caused it. So typically you have healthy men between the ages of, say, 18 to 35 who would try this potential drug, and what we're really looking for is, are we really hurting them severely? Are we causing organ failure? Are we causing death? If that first phase works out, you would then go to phase 2. At this point, you hopefully know. So phase 1 is done maybe in about 20 people. Phase 2 is done, say, in up to 200 people. And your goal is, okay, you now know that you aren't hopefully hurting too many organs, or you aren't hurting, affecting people's lives. In that situation, what you start doing in phase 2 is seeing what is the impact of something like this in terms of general safety. And what that means is, are you, even though you aren't actually hurting people in terms of destroying their organs, maybe you're causing nausea, or you're causing vomiting, or you're causing diarrhea, or you're causing heart murmurs, or whatever it is. In those situations, what you need to do is you need to make sure that you start recording that information. The only way to do that is by testing it in more people. So at this point, you still don't know if this potential product is effective. All you know is that we're hoping that it's safe. So then you do phase 2. At the end of phase 2, hopefully you get good results. You think that this product is still safe, that the risks outweigh the benefits. And if the risks outweigh the benefits, then you go to phase 3. Phase 3 is up to 2,000, and I've seen studies up to 65,000, 70,000 patients. Well, I keep calling them patients, but they are not patients, they are subjects. And the reason they're subjects is because you aren't treating anyone. The goal in a phase 3 study is to evaluate if your product is effective. And the goal to see if a product is effective is to see, does the potential drug do what you expect it to do? If you expect it to cure heart murmurs, does it cure heart murmurs? The fact that it also cures blood tumors, that may be irrelevant. You're only looking at it for the perspective of, does it do what you want it to do? If it also works to cure blood dyscrasias or blood tumors, in those situations you might do a separate study, but that's not the goal in a phase 3 study. The goal of a phase 3 study is to look at a single or multiple planned endpoints being goals. If all of these things go right, then you finish your phase 3 study and you are ready to go into go back to the FDA and say, look, we've done all of these things. We believe that our drug is both safe and effective. That's what the FDA is worried about. Is your drug safe? Is your drug effective? Phase 1 and 2 look primarily at safety. Phase 3 primarily looks at effectiveness. The FDA is also worried that your drug is either misbranded or adulterated. That's really more of a manufacturing process and a advertising or promotional thing, and we might discuss that in a future discussion. But as it relates to safety and efficacy, this process that I just described to you often is related to determining whether those goals are met.